Today I will be talking about transcription in a prokaryote cell. Transcription is a process in which information from a strand of DNA is copied into a molecule of RNA. In transcription, DNA is used as a template and no primer is needed. The RNA product that is made is complementary to the template strand and is called the coding strand. The difference between the template and coding strand is that RNA coding strand, all of the T nucleotides are replaced with U nucleotides. Now, let's break down transcription. Transcription has three main steps: initiation, elongation, and termination. First, in the initiation step, the DNA molecule unwinds and separates to form a bubble. To do this, a sigma factor causes RNA polymerase to change from a core enzyme to a hollow enzyme so that transcription can occur. RNA polymerase then binds to the promoter region, which is the -10 box, the -35 box, and the +1 region, as seen in the diagram. Furthermore, the sigma factor is added so that the promoter region can be recognized by the RNA polymerase, so it can bind and start to transcribe at the right place. Once RNA polymerase binds to the promoter of the template strand, it sends a signal for the DNA to unwind and forms a transcription bubble. Also, phosphodiester bonds are made, which will eventually add the nucleotides. Finally, the DNA strand is ready for transcription and will begin at the +1 nucleotide region as shown in the diagram. Now, let's say the -10 box is deleted in a gene that codes for an enzyme to metabolize sugar. RNA polymerase will not recognize the transcription unit, so that transcription will never begin and the sugar metabolizing enzyme will not be produced. Next step is elongation. RNA polymerase runs along the template strand and leaves the unwanted DNA strand behind. To start elongation, the RNA polymerase enzyme proceeds along the DNA template strand in the 5' to 3' direction, which is where the RNA attaches. Then the transcription bubble reads one base at a time and adds the RNTPs, which synthesize the RNA, and two phosphate groups are cleaved from the incoming RNTP, as the nucleotide is added from the phosphodiester bonds. Finally, the sigma factor leaves after it binds to the RNA polymerase, and we are left with the RNA strand that is identical to the DNA strand, except for the T nucleotide in the DNA strand are now U nucleotides in the RNA strand. Finally, termination occurs. In Rho dependent, the RNA contains a binding site for the protein Rho factor. The Rho factor binds to the 5' end and moves up the transcript toward the 3' polymerase. The Rho factor then uses helicase to unlink the DNA-RNA chain, and this causes termination. In Rho-independent termination, when RNA polymerase hits a region rich with C and G nucleotides, it folds back onto itself and creates a termination hairpin loop. Because of the termination hairpin at the C and G rich region, if there is a long sequence of A's on the template strand, it signals termination, so the RNA breaks off because it's a weak point. These are my references.